we're a scientific institution, which means we have an obligation and a duty to be at the forefront of this battle against the coronavirus, which means that we're trying and we are harnessing all of our scientific capabilities and resources, and we harness those in order to contribute. And we contribute in different levels. We try to make an impact in the short term. So very recently, we concluded the building of 2 facilities that would help conduct coronavirus tests in Israel. One of them in the INCPM, in the Institute for Personalized Medicine, in which we took one of the biological services labs that uses PCR machines. We increased the number of PCR machines, and we used several dozen volunteers, all of them students and staff from the Weizmann Institute, who would run coronavirus tests. We'll probably be able to go up to a few thousand tests per day, which will at least double the national number of tests which are being carried out. In parallel, Ido Amit and Eran Elinav, 2 of our brightest scientists, have come up with a new idea to expedite and make many more such coronavirus tests in parallel, and we've also established a facility that uses their method. In the more intermediate and maybe longer term, I think it's, for me, I come from the physics world. I'm a quantum physicist. And before I started this role, I knew very little about biology. And to me, it's amazing to see how many people across campus, biologists, chemists, computer scientists, physicists, are dropping their own research because they understand the urgency and the need, and they look for ways for them to contribute against in the fight against coronavirus. And there are various efforts. Some of those efforts come from the world of vaccines. Some of these efforts, like what Neil would tell you, come from the world of drug discovery and the effort of expediting drug discovery, which usually takes months and years, to a short time scale where we can have an impact on the status of coronavirus plague right now. So with that, I would like to introduce our speaker, Nir London. Nir's lab, which is named after the Sherman family from Canada, specializes in applying computational as well as experimental methods to find compounds, to find chemicals that can mitigate the effect of different pathogenic proteins. And I think a large part of these methods can be applied towards fighting viral diseases. And like many other scientists at the Weizmann Institute, Nir is now turning his sophisticated scientific machinery to try and find remedies to discover drugs that would be able to mitigate the effect of the coronavirus. And Nir is doing that in a very special way through an international collaboration, both with institutions in the UK as well as in the US. So Nir, please tell us about your journey.
Thanks, Ray, for the introduction. I would like to present a collaboration between several groups, unofficially headed by Professor Frank von Delft, who was also on the call, who initiated some of the work that progressed into this. And so let's get right to it. So I guess I don't need to introduce COVID-19, which by now is a global pandemic with over 500,000 confirmed patients and about 22,000 deaths so far, which, as Roy and Sheridan mentioned, did not skip Israel and the UK. This is a very dire situation, and everyone are trying to fight it. Just one slide on introduction about the actual agent. So here you can see electron microscope photo from a recent paper of the coronavirus. It's called corona because of these little spikes on its envelope that makes it look like a crown, since the name corona. A few numbers from a paper by Ron Milo at the Institute. This virus only has 30,000 RNA bases, which is a tiny fraction of, if you want to compare the human genome, which is 3 billion DNA bases. And these encodes less than 30 proteins. So little information is able to wreak havoc over our entire global civilization and economy. Once the virus goes near our cells, it only takes a virion, one little virus, 10 minutes to enter the cell. Then within 10 hours, it finishes a replication cycle. And at the end of the replication cycle, the cell explodes and releases 1,000 new virions, 1,000 new units. So this is why it spreads through our lungs, basically. We came to the conclusion that it must be stopped. In general, there are two main strategies to combat viruses. One very successful strategy is using vaccines. There are successful vaccines against viruses such as human papillomavirus, HPV, mumps, measles, and rubella viruses. All the population is basically vaccinated against these, and they're pretty much gone from the world. It takes a lot of time to develop vaccines, and it takes specifically a lot of time to test them clinically. But other consideration that not a lot of people are thinking about is their cost of manufacturing and their logistics. So vaccines typically need to be refrigerated in order to be able to be shipped around. So third world country, for instance, even if a vaccine will be generated in time, it's highly unlikely that many millions and millions of people in third world countries will not get access to such vaccines because of logistics and costs of goods. On the other hand, another strategy that has been heavily used to fight viruses are antivirals. And these are small molecule compounds, so chemicals, typically being taken orally by a tablet. Examples for successful antivirals are the many drugs that we have against HIV or AIDS, as well as hepatitis B and hepatitis C, which was recently developed. Other viruses are also have antivirals. These are typically cheap. They can be manufactured on a large scale. They're easy to ship around. And so our goal of the consortium that I'm about to describe is to develop a cheap and safe antiviral against this SARS-CoV-2. And specifically, we're targeting protease. It's one of the 30 proteins in the virus that is essential for its activity. Without this protein, the virus cannot reproduce. And we want to develop this cheap and safe antiviral faster than traditional drug discovery pipeline. Why is that? A traditional drug from conception to FDA approval typically takes between 10 and 15 years and between $1 and $2 billion. And we have neither, and especially we don't have the time. So our approach is to remove the barriers of traditional drug discovery. And specifically, drug discovery goes through a linear, iterative design, make, test cycle. So you think of some compounds, you synthesize them, then you test them in a cheap assay to see if they work, and you focus on small list that works, and then you do it again and again and again, linearly until you converge to something good, and then you only then go to the next step, which is typically more expensive. So that's this linear fashion takes a lot of time. It's cheaper, but it takes a lot of time. And the other thing is the bureaucracy, IP, and market considerations that plague traditional drug discovery. So to get rid of these, this entire project I'm going to describe to you is completely open science. No IP is attached. No one is going to get rich over any drug that will be developed during this project. Everything is shared in real time with the entire scientific community, and actually with everyone who wants to see it. It's available on the internet. And so this eliminates all the legal people, all the material transfer agreements, all the market consideration. Many drug development programs for viruses such as Ebola were terminated because they were just not financially viable. And so here, all of these are out of the table. The other thing that we bring to the table is some force multipliers that are unique to this consortium. The first is crowdsourcing of global intelligence, which I'll describe in a second. The second is unprecedented amount of preliminary data that we were able to generate and breakneck speed. Basically, within the last month, we generated an amount of data that can support a full drug discovery pipeline. And then the third is relying on machine learning and crowd computing. So let me just describe briefly how the past month looked like for us. So in February 14th, this main protease, which is the protein in the virus that we're targeting using our antiviral, was produced for the first time at Diamond Life Source, which is the facility that Professor Frank van Delft is in charge of. Within a week, they were able to determine the atomic resolution structure of the protease, which means that now we know exactly how the protein looks like, which is the basic entry point into discovering a drug that would bind in the active site of this protein. In parallel, they shipped samples of this protein that they generated to us at the Weizmann Institute. We were able to screen it using technology that we developed over the past couple of years. And very quickly, we found 130 preliminary hits, 30 of which were validated in a much more stringent environment. And since both we and the people at Diamond in Oxford had a copy of the library, they could in real time test the compounds that we found to be active. We, in the meanwhile, started working on synthesizing more elaborate analogs of the compounds that we found. Then, in a heroic effort, the team at Diamond were able to collect 1,500 crystals. So you can see these tiny crystals in this droplet. Each crystal is made out of this protein, and in each one there's a different compound. So they're trying to take snapshots of how does this protein looks like when our drugs are bound in the active site of the protein. And they were very successful, and this produced tens of structures. You can see here all the colored compounds are occupying different pockets within the protease active site. And now the challenge is to take this very preliminary compounds that are able to inhibit this protein and turn them into an actual drug. So as you can appreciate, since we have about 80 different compounds here, the combinatorial question of which compound might we be able to merge with other compounds is just exploding combinatorially. So in order to address this, we published this data set, the structural data set online, and we called to arms medicinal chemists around the world. We just told them, you know, here's everything we found. Please come up with designs for what you think we should synthesize and test next. And within a few days, we got hundreds of proposals from medicinal chemists all over the world, people that are working in pharmaceutical companies, people from academic institutions, actually some people that has no background in medicinal chemistry. And now the challenge is to take these hundreds of designs, and you can see a few of the very versatile designs. So each small structure here is a structure of a compound that someone suggested we should try to make and test against the protease. And so in order to prioritize these, we were collaborating with a company from the San Francisco Bay Area called Postera, which developed machine learning algorithms to prioritize which compounds are more suitable for synthesis and which one will have a better chance of working. We're also collaborating with Sloan Kettering on, they're using simulations of how will any of these drugs bind in the binding pocket of the proteins. They're using crowd computing, so people around the world are donating their computers for these simulations to run on their home computers. And so they're able to parallelize and basically prioritize all of this list in a very short time period. So just to introduce the team and and the methodology. So we started this with our covalent screening technology and synthesis of some original hits at the Weizmann Institute. Frank's team and the diamond light source determined all of those crystal structures that I showed you. Hundreds of people around the world have submitted already designs for next generation compounds. Postera, machine learning company in San Francisco, is prioritizing these. Then Sloan Kettering does the drug binding simulation. Inamine is a Ukrainian chemical synthesis company. They have offered to synthesize anything we will tell them based on this effort at cost, basically. And they can also do some preliminary testing of biophysical properties. Drug compound has to have some properties such as solubility. It has to be soluble enough. It has certain properties that would make it suitable for administration. And then they will synthesize and test all of these compounds on site. Then they will ship synthesized compound to various locations such as the UK and Canada for testing of these compounds for protease activity to show that these are in fact able to inhibit the protease activity. And then the best compounds will go further for collaborators in the University of Nebraska and Frankfurt University to be tested against the live virus. And so we want to do all of these tests instead of in the traditional way that drug discovery works linearly. We want to do all of this in parallel. So instead of following up only on a few tens of compounds, we aim to follow up on 500 to 1,000 different compounds in parallel, and so drastically shorten the time scale that it would take to find that one compound that might work. And our premise is that if there is a safe compound, something that's safe to take and shows efficacy against the virus, humanity needs to know about this fast. So as I mentioned, we're trying to do everything in parallel, and basically no traditional funder would back such an effort because it breaks all the paradigms that are so far held in the drug discovery field. And so we're looking, the more funding we'll get to drive this project, the more compounds we can test in parallel, and then the shorter the time frame would be to get to an effective drug candidate. And we hope, our aim is to come up with something in the next few months in a time frame that would be relevant before this pandemic become really catastrophic. I should mention that there are other efforts ongoing in the world. Some efforts target other proteins, not the protease, but for instance, you may have heard of a spike protein that allows the virus entry into cells. Some groups are working against that. Other groups are working against a polymerase, another essential enzyme in the virus. And there are actually other groups also working against this protease. I think all of these are complementary to our efforts. As previous antivirals showed, a cocktail of drugs is sometimes much more efficient than targeting only one specific protein. And so I wish all of them great success. And I hope that we can contribute an active molecule that might have an effect on this virus. At this point, I want to thank you for your attention, and I want to please let Frank give you his perspective on this project.
Hi, I'm Frank, and I should say Nira's covered it very well. I'll just say two things. The first thing is that this has been quite overwhelming. This facility that we started at Diamond several years ago, it's always been clear that we can generate the kind of data that is not exploitable even by algorithms at this point, and should in principle be able to achieve a real leap from first experiment to drug candidate. It's never been clear how to do it though, and people around the world are working on algorithms. And this collaboration we actually assembled in 1 week. So that slide you saw on the world chart was 1 week of thinking. I've never been part of something like this before. I've been part of many large collaborations, but nothing that just crystallized so quickly. It's been truly overwhelming. Also, the prospect of tapping into so many brains across the world built up over years of investment in many companies and many projects. This collective experience now being on tap for something like COVID is quite remarkable. So I keep having to pinch myself that this is even conceivable. Whether we'll succeed or not, as Nir said, we don't know. We have to try. We have no choice. We have to try. We're sitting on we're not sitting on, we've got this pile of data which surely needs to be able to contribute. Everybody says drug discovery is hard and it keeps failing. Well, we may fail as well, but we have no choice. We absolutely have to try. Second thing is just to give you a sense of this parallelism that Nir described. There's a image that I like to invoke about this. Usually if you do drug discovery, you are trying to roll a ball down a hill along terraces. So you'll kick the ball down and you see if it bounces downhill. If you hit the terrace, you have to find where the next downhill is, and you kick it again. And then eventually, after a few years, you may be at the bottom of the hill, or let's say the bottom of the funnel. What we're trying to do is we want to play golf. There's a big open expanse. Somewhere on that open expanse is a hole. And we want to get the ball in the hole. And now you could keep hitting the ball around your field, and a few years later, it might fall in the hole. We're going to try and hit many, many balls across in all directions and hope that one of them falls in the hole. If it falls in the hole, we will know it. And that ball that falls in the hole will be this compound, which will be safe, effective, and crucially cheap to make. So that's something that doesn't tend to get put into the pipeline. So yes, and beyond that, I thank you for your attention.
Thank you so much, Nir and Frank. That was absolutely fascinating to see how real science is working now and that kind of amazing collaboration. Somebody has asked, how can people donate their computing time or power to the project?
The drug binding simulations are happening at a project called Folding at Home. Mm-hmm. So if you Google Folding at Home, you'll be able to register your computer to this global effort. And in fact, I just read yesterday that Folding at Home became the world's largest supercomputer. So it's actually 10 times faster than the previous largest supercomputer, just by the fact that so many people want to contribute something and donate their personal computer cycles to this effort.
A question that I received earlier, which is maybe a bit trickier, but a few people have asked me, do they really think that the institute is best based to be producing drugs to do this rather than the pharmaceutical companies, given that normally our focus is on basic research?
Right. So I myself came to this project from a basic research sort of perspective. So the main question we're interested in our lab is, how do small molecules bind to protein? And we're developing specific technologies to address this question. The reason I know Frank is because we collaborated for the past 3 years on specific types of proteins that are suitable for our approach. And when COVID erupted, this protease protein that we chose out of the 30 proteins that you mentioned is very suitable. So I don't want to get into the scientific details, but the active site has this unique amino acid called cysteine that our technology can target really effectively. And so that's how we got into this, and it worked better than we expected. So I agree with you that the Weizmann Institute is not the best institution to run clinical trials, but since we were able to contribute such crucial starting points for this process, and we're basically now it's out of our hands. So the rest of the experiments are not actually being done at the Weizmann Institute, but rather compounds are being synthesized somewhere else, and we're now more coordinating this effort and sort of driving it forward.
Thank you. How many of the 30 proteins are essential, this person asks?
I'm no virologist, I have to admit. I'm a chemist and a computer scientist. I know of at least 3. So this protease we're working on, the RNA-dependent polymerase, and the spike protein that's responsible for the virus entry. Maybe Frank knows a little bit more.
I'll just say, in general, viruses are very parsimonious. They have evolved to be as compact as possible. So in general, all the enzymes that they encode for tend to be necessary. And you can, for instance, as a lab tool, you can make a virus that has a reduced genome, they call them replicons, might have some of the functions of the virus, but can't complete the life cycle. So one could, in principle, target any of these, or in fact, multiple ones is usually a good strategy if one has time. But there's always a practical question, is which of these proteins are you able to produce in the lab and study in depth? And we were lucky that the Chinese moved extremely quickly for this particular one, and they had the structure solved back in January. What Nir described earlier was the reproduction of the work in Oxfordshire. So we happen to have this reagent in hand, and we could build on that. But in principle, in fact, we are currently, Diamond Light Source is reaching out to groups around the world to bring any other structures that proteins they've already worked on to date. And there are now multiple ones, at least a half dozen, that we want to work up as well and really add to the pile of information that's known.
Thank you. I have a few more general questions from people, maybe not necessarily specifically related to your research. Somebody has asked when we think you might have an antiviral ready to administer to patients.
We hope month, but we're aware that this is, that's what we call it, a moonshot. It's highly unlikely, but that's our goal.
And then I have an older question that someone wrote to me about earlier this week about there's been lots of reports about chloroquine, and do you know of any other drugs that have been tested already and therefore are safe in human? Have you heard of any other promising things that could work?
So there were conflicting reports about chloroquine. There was a very promising study, clinical trials, a small number of patients, but actually, surprisingly, the scientific community later dug into the data, and it seems like it's less promising than it was originally touted. So I think there's no strong evidence that chloroquine is really effective, but there's maybe enough evidence to try and pursue a bigger trial. They started a trial with an antiviral from Gilead that was originally developed against Ebola. So that sounds promising, but as Frank mentioned before, this is a really complicated drug, and it will probably be difficult to produce en masse. So it might be useful. We'll have to wait on the results. But so that's another direction. And the third direction I'm aware of is HIV protease inhibitors are currently being tested. The state of Israel, for instance, allowed generic use of HIV protease inhibitors for doctor to administer to patients. I don't think there's any clinical proof that they're effective, but there's a lot of logic to think that there will be.
I will add to that for all the current ongoing trials, and there are many obviously, the premises tend to be not very strong biochemically. So there seems to be a reason to try it, and if the doctors have nothing else, they will go for it. But the very, very hard evidence that this will be effective is hard to get, not least for practical reasons. So the other thing that is happening with increasing speed is that people are trying to test in a broad way compounds that are known to be safe in the clinic, and also drugs, but also things that have failed drugs, but they failed because they just didn't work for what they were designed to work against, and to try and find if anything falls out from there. All of this is extremely promising, and it's being looked very hard at by people that are much better at it than we might be. But they have one potential stumbling block down the line, and that's the cost of manufacture. If they are complicated to make, scaling them up, getting them out to a large population of people that are going to be infected will be tough. And that is really the main reason we even thought it's worth embarking on this, working with Alpha Leaf from Post Era, that we can bring in this question of synthesizability at the very start. That's our key requirement. The other things are necessary, but that's the key one. So drug repurposing is clearly promising, and well, we hope it works, but we don't know.
Thank you. A few more questions have just come in. Somebody has asked me, how are the results from all the research in parallel reported and acted on? Is there a special platform used by the scientists to see the results and to go through the admin and coordinate? Indeed, can you show a screenshot that shows the global reporting and analysis? I'm not sure if you can do that.
It is an excellent question. It's the one that worries us most, because as I said, this is a week-old project. There's nothing that we can just turn to and use. We have to make many things up. In fact, Postera within a few days set up this idea harvesting website, which was sensational. We are now that we've got the number of things set up, looking at the project management infrastructure we can use. There are things we can probably deploy. And these are, our actions are live. We have daily discussions every late afternoon from me to see what's the next most important thing to sort out. Mm-hmm. And so a design and management platform is crucial. And we probably will have something online by next week when we have the first data of the iteration start materializing. Yeah. But anyway, if there are ideas floating out there, we'll certainly have our ears wide open. But I think what drives it all is that whatever platform we use will always be fully visible, and that is key.
Yeah.
Can you see my screen?
Yes, thank you.
Well, so you see, this is the website that Postera hosts, where people can come in and just draw their molecules straight here. Wow. Submit them. Give the design rationale, notes, any files on models they made out of these, which fragment they were based on. And then once they submit it, you can see here all of the design molecules submitted by everyone. So you can take inspiration from other people's designs. So if we just can go into one of them and see more details. And also, each one of these designs has sort of a forum entry around it, so people can discuss between them, talk about the strengths and weaknesses of each design. And as we now, so the first batch, the first stage of this challenge ends at midnight tonight, and then we'll send the first batch of compounds to synthesis. And then we'll report back results using this platform. We'll report back results. This is the structure of this compound, and this is how much it worked. And then they can keep designing the next stage of compounds. And as we actually, during the last week, what was phenomenal is that people have actually approached us saying, we're a chemistry lab, we can synthesize compounds for you. So maybe we can't contribute designs, but we can contribute synthesis. And some biology lab approached us and said, well, we can't synthesize compounds, but we can test them against the protease. We have the protocol. So we're thinking now of crowdsourcing even more with having, people could claim designs and say, I'm responsible for making this compound, and then they'll ship it to, probably will center everything at Enamine, which is the chemical vendor. So they will do quality assurance of everything that gets synthesized, and they'll be the logistics center. So chemists around the world could send compounds to them, and then they will redistribute them to biologists that would test them. And then we'll sort of take this crowdsourcing to the next level, not only at the level of ideas, but actually at the levels of chemistry and biology.
I've had a question about why is it taking so long to find a test that lets us know if we've had it?
As far as I know, there are, at least in the US, probably other places around the world, there are tests now that can take as little as 15 minutes, like a home pregnancy test. 500,000 of people are already infected. Probably many millions are already infected and don't know about it. And all the countries are scrambling to get their hands on reagents that will enable such tests. So I think shortages and logistical problems that are because of shutdowns at different places complicates the logistics of all of this.
there's also one scientific reason. It's that the virus hasn't been around long enough to even be able to establish that. The experiments you need to do are kind of in the order of months, and that's how long it's been here. So to know whether it's effective is really tough. And so you have to try and model things or infer. And so if you then start making a test available and people trust it, but it's not completely clear, that could almost be worse than just sitting back for a while. So it's tough. It's a really, really difficult problem.
I am going to wind this up now. Luckily, someone's asked a question that is close to my heart. And somebody has said that you mentioned that there was maybe a shortage of funding, and where could we donate to? So I would like to first of all thank Frank and Nia very, very much for a properly fascinating talk. It really was amazing to listen to. I'd like to thank the Institute for putting this on, and the small team in London under Sheridan, who have done a great job revamping our website. So to the person who asked that question, if you go onto Weizmann.org.uk, you can donate directly to the coronavirus project or thing that the Institute has put up. It has created. So it doesn't just have to go to general things, it can go specifically to the coronavirus work. And related to this, I'm pleased to say that the trustees of Weizmann UK agreed today to give $1 million to that fund from our reserves to kind of kick it off. So we hope that will help you. We are very grateful for everything you're doing for us and the world, and good luck, and thank you very much.